Hello, my name is Samoda Fernando. I'm an assistant professor in the Department of Animal Science at University of Nebraska-Lincoln. Today I'm going to talk about rumen microbes and fermentation. Many of you might have heard that the microbes within us play an important role in obesity, diabetes, inflammatory bowel disease, and many other diseases. But if you compare a ruminant to a human, the role of the rumen microbes is far more important than a human. The reason that ruminants can survive on low quality forages is because of this rumen microbial population. These rumen microbes actually provide the animal with nutrients that are not available to the monogastric animals such as you and I. But microbial fermentation or microbial digestion of cellulose is a slow process. Therefore, the feed has to remain in the gastrointestinal tract for a long period of time. So to facilitate this, the ruminant animal has developed a four-chambered stomach, which includes the reticulum, rumen, omasum, and abomasum. Among these four compartments, the rumen is the largest and accounts for a majority of the digestion. So if you look at the role of the microbes towards the animal's health and performance, about 70% of the energy that the animal gets is from microbial breakdown. So there is a very mutualistic relationship between the microbes inside the rumen, where the host gives an environment for these microbes to grow and thrive, and these microbes in turn provide the animal with energy for growth. And we know that diet actually is a very large contributor of how this microbial community is affected. So diet is a factor that actually changes or contributes towards the composition of this rumen microbial community. So what's the rumen? We can consider the rumen as a fermentation vat. It's anaerobic in nature. What I mean by anaerobic is it's free of oxygen. So the microbes that live within the rumen live under conditions that are free of oxygen. And these microbes are designed or are very efficient in digesting cellulose-rich diets and provide the animal with nutrients in the form of volatile fatty acids. Or these microbes itself could become microbial cell protein and be a protein source to the animal. The rumen can hold between 40 to 60 gallons of rumen fluid and accounts for about 60 to 65% of the gastrointestinal tract of the animal. If you think about the rumen microbial population, it's a very complex microbial population, and all three domains of life is actually found within the rumen. So we see the bacteria, which is the major component of the rumen microbial community. We also see eukaryotes, which fungi and protozoa belong to, and we also see archaea, some of the methanogens, and we also see viruses within the rumen. So the picture on your top right hand side that you see is actually a picture from the rumen where you can see this complex microbial environment. You can see this large protozoa within the rumen environment and bacteria actually teeming within this on this background. And you can also see these bulbs that you see on this protozoa is actually sporangia or some of the reproductive structures of fungi, depicting that there's fungi actively growing inside the rumen as well. And you can see this is a feed particle that you can see the outside. When it's intact, the microbes can't attack it, but once it's broken down, it actually gets colonized by the microbes and gets digested. So if you look at the microbial population within the rumen, the bacteria are the most abundant population. They account for about 80% of the total microbial community, and about 75% of these microbes are actually attached to feed particles and are actively digesting the feed. There are about 25% of microbes that are free living, that actually are motile and can be moving towards different substrates. The second largest population inside the rumen is the protozoa. These protozoa are known to depolymerize pectin, help engulf and metabolize starch granules, and these protozoa can actually feed on bacteria as well and help control some of these bacterial populations. Anaerobic fungi have also been isolated from the rumen, and anaerobic fungi are only found in a very few environments. And these fungi inside the rumen actually help solubilize lignin, or they synthesize these lignin modifying enzymes that help bacteria get access to feed particles to digest this lignin rich feed or low quality foragers. Archaea and viruses have also been found in the rumen, but we know very little about these populations. But archaea, the methanogens or the methane producing microbes that you find inside the rumen, belong to this domain archaea. So what's unique about the microbes that's found in the rumen? So the microbes that's found in the rumen has these unique structures that help them actually get energy from these low quality foragers or from the diet that these animals are being fed. One such structure that you see on your left hand side is called the cellulosome. So a cellulosome is a structure, it's a multi-enzyme complex, so it has multiple enzymes, but it also has these unique domains, or we say carbohydrate binding domains in it. So what this cellulosome does is actually it binds to these polymers or complex carbohydrates that this animal feeds on, and then using this cocktail of enzymes or this multi-enzyme complex, it can digest these cellulose rich or very complex carbohydrates and then absorb these oligomers or monomers and then utilize them to get energy from them. So what about microbes when we have in a finishing diet and when we have a starch-rich diet? Some microbes have structures known as these special binding proteins that bind to starch molecules. And by binding to these starch molecules, they can attract them towards them and then digest these starch polymers to gain energy from these molecules. So these microbes that you find in the rumen have adapted or have unique structures that help them utilize these low quality foragers or to utilize the substrates that the animal eats. But if you think about the rumen microbial community, it's actually a very complex community with lots of interactions. So if you take the microbial community, each individual species doesn't have all the enzymes needed to digest the wide variety of substrate that the animal eats. So what happens is there is a microbial food chain where a byproduct or an end product of one organism might be an substrate that a different organism can use. For an example, if you look at this complex polymer or complex carbohydrate or the plant cell wall, there are certain microbes such as Ruminobacter or Fibrobacter species or different microbial species that has these structures like the cellulosome that can bind to this complex plant cell wall and break down it into its monomers, or we call it or oligomers, that means short pieces of the carbohydrate. And the short pieces or the monomers or individual carbohydrate molecules can be then utilized by other microbes and provide volatile fatty acids. And in the process, they will produce CO2 and hydrogen, which methanogens could utilize and make methane. So there is all these interactions that actually make the rumen ecosystem efficient because of these microbes working together to make or enhance the amount of energy you can get from these low quality foragers. So to give you an example how these microbes work together, we look at fungi. So I mentioned earlier that fungi are the only microbes that actually have delignifying enzyme or lignin modifying enzymes that could remove the lignin. So if you look at your right hand bottom picture, what you see is when fungi, the cartoon shows that when fungi attach, the lignin is removed and now the microbes have access to cellulose and other polymers that they can digest and get energy from. So if you look at the graph that you see here, you can see when you have an intact forage with lignin, the amount of cellulose remaining is way higher than when it's delignified with fungi. So your digestibility increases. So these fungi actually help especially on these mature foragers, low quality foragers, which has lots of lignin to help solubilize this lignin and help microbes attack those foragers. So what happens to these monomers, or once the microbes use their enzymes and break those polymers down to its basic component, such as glucose, what happens to this glucose? So this glucose is then utilized using glycolysis and different pathways to actually produce different volatile fatty acids. So we know that the three major volatile fatty acids that's produced within the rumen are acetate, propionate, and butyrate. But in addition to acetate, propionate, and butyrate, you could have lactate, ethanol, and other substrates as well. And if you look closely at some of these reactions, you can see when we produce acetate or butyrate, we are actually producing CO2 and hydrogen or carbon dioxide and hydrogen as byproducts. Whereas if you look at propionate production, we are actually producing CO2, but we are utilizing or using some of the hydrogen. So these byproducts are actually what other microbes, such as archaea or methane-producing microbes, actually use this hydrogen and CO2 to produce methane. So some of these byproducts are removed from the system from these methanogens and other organisms, such as by converting it to methane or other intermediates. Say, for example, when you have lactate being produced, there's microbes that can convert this lactate back to propionate. So there is this network or a connection. There's a lot of interactions between this microbial community that makes these the ruminant animal or the rumen to be efficient in utilizing these low quality foragers to provide energy to the animal. So what happens to the VFAs once it's produced in the rumen? They get absorbed and these by the animal through the rumen epithelium, and once they are absorbed, the animal actually utilizes the VFA, and using the TCA or tricarboxylic acid cycle, it can actually utilize those substrates to get energy from those volatile fatty acids. So in a nutshell, the microbes within the rumen play an important role in nutrient digestion. So about 70% of the animal's energy actually comes from microbial breakdown. And as I mentioned earlier, these microbes itself can actually flow to the small intestine and become microbial cell protein and be a protein source to the animal. Therefore, microbes are not only helping the animal get energy from the diet, but themselves could become actually a protein source for the animal as well. So therefore, these microbes play a key role in nutrient digestion within the animal. Therefore, the rumen microbial population directly impacts the performance of the animal. So if we can better understand this rumen microbial population and how we can use diet and other factors to change the microbial community, we can actually have an opportunity to increase animal performance, increase animal health, and there'll be many benefits of by actually manipulating the rumen microbial ecosystem that the animal can gain. Thank you.